Hello everyone, welcome back to my YouTube channel. Today's video is all about the structure of bacterial cell. So structural components of bacteria are as follows. One, cell wall, second, cytoplasm, then flagella, pili, fimbriae, and this is the structure of bacterial cell. So you can see here in this structure, first that is a plasma membrane, then cell wall, sorry, capsule, okay, then fimbriae, cytoplasm, entire ribosome, nucleoid, inclusion bodies, plasmid, and this is a flagellum and pilus. Right. Now first we discuss the cell wall. So as we know, cell wall is the outer tough covering around the cell. Okay, cell wall provides specific shape and protection to the bacterium. It's also function to cell wall prevents the cell from swelling and bursting lysis due to the osmotic changes. So despite their common function, the cell wall of bacteria and eukaryote are structured very different. Now the glycocalyx are sugar coat or sugar shell. So what is glycocalyx? The glycocalyx is a layer of glycoprotein, that means glucose plus protein, and glycolipid, that is glucose plus lipid. Okay, that means carbohydrates plus lipid and carbohydrates plus protein. That surrounds the cell membrane of many types of cell, including animal cells and bacteria. It's often described as a sugar coat or sugar shell due to its composition of sugar molecules, that is carbohydrates. Right? It is composed of polysaccharide uh, called the murin. Okay, murin, also known as the peptidoglycan, is the polymer that forms the rigid cell wall in bacteria or peptidoglycan. Okay, so in structure, you can see in this structure. Okay, so here it is the this is a peptidoglycan, right? Entire structure is peptidoglycan. So here NAM, okay, NAM. It consists of polysaccharide NAG and NM and cross-linked. Okay, here it is cross-linked with short amino acid chain. Okay, it is a 4 amino acid chain. Okay, either the alanine, glutine, okay, leucine, mabylate. The structure of peptidoglycan of E. coli. This is okay, peptidoglycan of structure of E. coli. So changes consist, as I said, alternative NAG, NAM. Okay, residue joined by the both residues. Okay, NAG, NAM are joined by the beta 1,4 glycosidic bond. Okay, here it is the NAM and NAG. Okay. So parallel chain are cross-linked by tetrapeptide. Okay, 4 peptide, 1, 2, 3, 1, 2, 3. Okay, amino acid, that is a tetrapeptide. That's why I say. Okay, very in different species of bacteria. Okay, okay, this is forming, where they forming like NAM, 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 NAM. All the time that NAM, NAM with the tetrapeptide and link to the each other. Okay. The cross-link structure, peptidoglycan form a strong covalent, here it is the covalent shell around the entire bacterial cell. Sorry. Okay. Now the interestingly, the unique structure of bacterial cell wall also makes bacteria helpless to some antibiotic. Okay, uh, like penicillin and cephalosporin, and prevents the cross-linking of polysaccharides and inhibit the bacterial cell wall formation. Right. So penicillin. Okay, so this is structure of penicillin, beta-lactam ring, and cephalosporin. Both are responsible to the prevent the cross-linking of polysaccharide tetrapeptides and inhibit the bacterial cell wall formation. Okay. So penicillin, for example, inhibits the enzyme responsible for forming cross-linking between different strains of peptidoglycan. Okay, one is the here peptidoglycan strain, another one is here, and this is the tetrapeptide, and this transpeptidase. It is responsible for the forming the tetrapeptide. Right. So what penicillin does here? Penicillin. Okay. So this is the penicillin structure, beta-lactam ring. Okay. So, okay, so now understand how penicillin and cephalosporin inhibit the bacterial cell wall synthesis step by step with molecular structure and enzymatic action. Okay, so normal peptidoglycan synthesis in bacteria. Okay, structure peptidoglycan monomer, each repeating units. Here it is contained NAG, NA, attached to NAM, a short peptide chain like this one. Okay, NM short peptide chain, alanine, glutamine, lysine, alanine, alanine. Right. Now, this cross link with the another NM. Okay, right. Now the D-alanine terminal peptide, sorry, dipeptide of one peptide chain is cleared by the enzyme. Which enzyme? Transpeptidase. Okay, so this cleavage release one D-alanine allows the formation of peptide bond with another nearby chain. Okay, one alanine is released and they forming the bond between two chains. Now, what's the mechanism of penicillin, cephalosporin, beta-lactam antibiotics? Okay, so penicillin and cephalosporin have four-member beta-lactam ring. Okay, which is structure is similar to the D-alanine. Okay, which is responsible for the joining or cross-linking to the peptide. Right. So same, its structure is similar to the D-alanine. Now, in irreversible inhibition is occur here. Penicillin, so this is penicillin binds to the active site of transpeptidase enzyme. The enzyme thinks it is acting on D-alanine, but the beta lactam ring opens and form covalent bond with the enzyme. The enzyme activity is blocked by the penicillin. Okay, the inactivates the transpeptidase permanently. Here this is permanently transpeptidase inactivate. Okay, so this is the how penicillin works onto the preventing the cell wall synthesis in the bacteria. Okay, now mechanism of lysozyme. Okay, lysozyme. So lysozyme digests the bacterial cell wall by breaking beta-1,4 glycosidic bonds between N-acetylmumaric acid and N-acetylglucosamine. Right. So here is the lysozyme. Break down the uh, bond between two NM and NH. Right. Okay. So the cell of gram-positive bacteria is much thicker and contain less lipid as compared to the gram-negative bacteria. Okay. Now comes to the next cell wall of gram-positive and gram-negative bacteria. So bacteria are classified into gram-positive and gram-negative based on their reaction to gram staining. Gram staining is a technique introduced by Hans Christian in Hans Christian Gram in 1884. Okay, so I briefly explain the gram staining procedure. Okay, bacteria is first stained with a crystal violet dye. One dye is used first step. Second step, they are then treated with iodine, which is formed complex with the dye inside the bacterial cell. Okay. Third step, decolorizing agent decolorize is applied, like usually use the alcohol and acetone. Now, gram-positive bacteria retain the crystal violet stain and do not get decolorized. Right? They appear dark violet to purple under the microscope. However, gram-negative bacteria lose the crystal violet stain because their cell wall structure allows the dye to be washed out. Okay. They are then counterstained with the safranin, which makes them appear pink or red under the microscope. So this is the structure of gram positive, gram negative bacterial cell wall. Okay. So gram positive bacteria, cytoplasm, gram negative bacteria also here the cytoplasm. Here is the plasma membrane, both is the same plasma membrane. Okay, inner plasma membrane and inner, right? Now, here difference gram positive bacteria having the teichoic acid. Okay, teichoic acid. This is the peptidoglycan cell wall, and then here this is teichoic acid. Okay, okay. So teichoic acid embedded within the peptidoglycan layer or attached to its surface are teichoic. Okay, they can embedded like this on a surface also. Now another lipoic acid. Okay, this is a different, slightly different. These are encoded into in the cytoplasm. Okay, here it is a cytoplasm like this. Cytoplasmic membrane. Okay, so see the cytoplasm and cytoplasmic membrane here too. Okay, and extended into the peptidoglycan layer, and they may be commonly linked to the cell wall. Yeah. Here it is the gram negative bacterial cell wall. Okay, so inner membrane same as it is gram positive. Periplasmic space, they contain the peptidoglycan cell wall and another layer of outer membrane of another layer of lipopolysaccharides in the gram negative bacterial cell. Okay, so gram negative bacteria have the thin peptidoglycan layer. Okay, it is a thin peptidoglycan layer, and here is a multiple layer. Okay, so layer but contain high lipid content in their outer membrane. It is the lipopolysaccharide, that is the lipid plus saccharides. Okay, carbohydrates. So that's why the decolorizing agent dissolve the lipid, allowing the dye to escape. Okay, that's why the gram negative bacteria shows the pink or orange color, and gram positive bacteria show under the microscope purple color. Sorry. Okay. Now the comparison between gram positive and gram negative bacteria. Right. Grams characteristic gram staining reaction. Gram positive bacteria retain crystal violet dye and appears dark violet or purple. Right. Two. Uh, okay. Gram negative bacteria can't retain the crystal violet, get decolorized and appears pink or red after counter-chemical reception. Okay. Peptidoglycan layer thick, multi-layer, or here it is the thin. Teichoic acid present in the cell wall, and here it is absent. Teichoic acid, outer membrane absent but present here in gram negative bacteria. Lipopolysaccharide content virtually known in gram negative bacteria high and form outer membrane that acts as endotoxin. Flagellar structure, 2 rings in the basal body, 4 rings in the basal body. Okay, we will discuss later in this session. Okay, the flagellar structure. Toxins produced mainly in exotoxin, and this is the endotoxin. Right. Cell wall disruption by lysozyme, highly susceptibility. Okay, as peptidoglycan is easily broken down. Less susceptibility due to an outer membrane that protects the peptidoglycan layer. Now, susceptibility to antibiotics. Highly sensitive to penicillin and antibiotics targeting peptidoglycan synthesis, and it is less sensitive. Why? Because outer membrane acting as a barrier. Resistance to drying, okay, it is high and is low. Okay, so this is the on the cell wall of gram positive bacteria. Okay, so difference between lipoic acid and teichoic acid. Okay, it's only in surface or embedded slightly into peptidoglycan. Lipoic acid is cytoplasmic membrane through extended through the peptidoglycan to outer surface. Okay, so some of the example of gram negative and gram positive bacteria. So one thing you have to notice here, all the bacteria name, okay, scientific word. If you written in anywhere in scientific word, so always underline the scientific word, or you can make it in italics. Okay, So it is italics version. Okay. Staphylococcus, Streptococcus, Clostridium, Bacillus. Okay. These are the gram-positive, gram-negative bacteria example. Now next it is cytoplasm. Okay. So cytoplasm is a semi-fluid ground substance enclosed by the plasma membrane. In bacterial cell, particularly we discuss the bacterial cell. So which is phospholipid bilayer. Okay, cytoplasm cell membrane, phospholipid bilayer, but there is no cholesterol and other steroids present in bacterial cell. Cell membrane. So cytoplasm is appears granular. Why it is granular appears? Because it having 20,000 of 78 type of ribosome. Okay, that's why it's appearing like granule, which may occur singly. Okay, ribosome may be singly or they in cluster form. If they call clustered form, then we can call the polyribosome. Okay, so apart from the nucleoid, this is the nucleoid, right? Apart from the nucleoid, certain bacterial cell contain extra chromosomal genetic material called the plasmid. Here it is the plasmid. Okay, extra chromosomal DNA. The plasmid DNA replicates independently. It contains genes like fertile factor, F factor, resistance factor, and nitrogen fixing gene, NIF. Some plasmid may temporarily become associated with the nucleoid. Okay, it is temporary, not permanently. Sometimes it's become associated with the nucleoid. This one is, and called the episomes. Okay, it's known as the episome. So who is given the name of plasmid? Liderberg and Hayes. Each plasmid is circular. Every plasmid is circular, supercoiled, and double standard and necked DNA. Okay. Okay, so I written here also. Bacterial flagella. So bacterial flagella are hair-like appendages that provides motility of bacterial cell. Unlike eukaryotic flagella, which have a 9 plus 2 arrangement microtubules made up of tubulin, bacterial flagella are composed of single filament of the protein flagellate. Okay, so structure of bacterial flagella. Okay, so now we can see here is the structure of flagellum. Okay. So the bacterial flagellum consists of three main parts. First, that is the basal body, the most complex region. Anchor the flagellum to the cell membrane and cell wall. Act as a motor that drives the flagellar rotation. Structure vary based on gram staining. As I discussed earlier, that gram negative bacteria having the 4 ring and gram positive bacteria having 2 rings. So here it is the S M P L, the 4 rings. So this is the structure of gram negative bacteria cell wall. Now come to the hook. Hook is a cow shaped, okay, like cow shape, short structure that connects the filaments to the basal body. Okay, so filaments connects to the basal body, this function, and composed of single type of flagellin protein. This function is a universal joint allowing the filaments to rotate efficiently. Now third part, that is the filament longest portion, okay, a long wave like helical structure that extends outward from the cell, composed of 8 vertical rows like this: 1, 2, 3, 4, 5, and another side also 3. So size 20 nanometer in diameter and 1 to 70 nanometer in length. Responsible for genetic propulsive forces for bacterial movement. So this is the difference between the gram negative, gram positive flagella. It is the 4 ring you can see here, and it is the only 2 ring. Now, types of bacteria based on flagella origin. Bacteria can be classified into different types based on the number arrangement of flagella. Right. So first type Atrichos, no flagella. Some of the example of bacterial cells. Monotrichous, single flagellum at one end. We also see the structure in next slide. Okay, Vibrio cholera, Bacillus. Amphitrichos, one flagellum at each pole. Okay, Nitrosomonas, Cephalotrichos, multiple flagella at one end. Pseudomonas fluorescens, this is the example of bacteria. Lophotrochus, cluster of flagella at both ends. Spirulina volutans, example of bacteria. Peritrichos. Okay, so flagella distributed all over the surface. Escherichia coli, Clostridium tetani. Now we see this with the structure of flagella with bacteria. Okay, so Atrichos, you can see this is the no flagella, Lactobacillus and Paschuella. Second, Monotrichous, the example of Vibrio cholera. You can see in this one flagella, only one. Okay, it is Amphitrichous. So Amphitrichous, that is the one flagella at each pole. Okay, Nitrosomonas. One, okay, each pole. Okay, so many flagella are present in the Nitrosomonas. Cephalotrichus, multiple flagella at one end, only one end. So Pseudomonas aeruginosa. Okay, see here this flagella. Only one end, either this side or this side. Lophotrichus, a cluster of flagella at both ends. This is Spirulina volutans. Okay, it is the both end cluster of flagella. Peritrichus, flagella distributed all over the surface. Escherichia coli, and the name is a Clostridium tetani. Okay, here you see this flagella. Okay, here also, but is zoom out version. Okay, here also flagella. Now next is structure of bacteria, that is pili, or we can say the fimbriae. Okay, so pili or fimbriae are extremely minute hair-like structure. Pili in plural and singular we can say the pilus, and fimbria in plural and singular we can say fimbria. Thin hair-like protein structure that project from the surface of bacterial cells, especially gram-negative bacteria. Right. Now structure of structural characteristics. Size, fimbria are shorter and thinner than flagella. Okay, diameter range from 3 to 10 nanometer. They are more numerous than flagella, can be 100 per cell. Composition, both pili and fibri are made up of protein, that is the pilin. Okay, subunit of pilin. Unlike flagella, they are non-helical and non-motile. Okay, pili is non-motile. Now the function of pili and fimbriae, attachment and colonization. First function. So fimbriae helps bacteria adhere to the surface. Okay, surface like host tissue, important infection. Okay, and medical devices, catheters, implants, each other attach and form the biofilm. Okay, so this attachment and colonization is essentially for initially colonization of host cell and inhibition. Second function, conjugation, sex pili. Okay, pili called sex pili, where F pilus, also okay, you can say the F pilus is only found in male donor bacterial cell. It forms the bridge-like structure called the conjugation tube. Okay, like this tube, and this bacterial cell, and here it is another bacterial cell. This is the conjugated tubes. Okay, during the conjugation. Through this tube, the donor transfers genetic material, usually plasmid, to the recipient cell. Okay, this process, this entire phenomena, we can also say a mechanism, we can say the horizontal gene transfer in bacteria, including the spread of antibiotic resistant genes also. They also spread the antibiotic gene resistance. So the comparison with the flagella, so pili and flagella, okay, difference. So the diameter is different, 3 to 10 nanometer, here is 20 nanometer. Length, short, long, 1 to 70 micrometer. Number may be many per cell, hundreds. Okay, few, one or few cells. Okay, per cells present flagella. Function, attachment, conjugation, and motility. Composition, pili protein, flagellin protein. Here is flagellin protein. Motile, non-motile, and motile. Okay, so this is the all about the today's video about the structure of bacterial cell. Okay, thank you for watching, and don't forget to my YouTube channel. Okay, don't forget to subscribe my YouTube channel and also share this video. Okay, thank you very much.